Tracing neural connections, a methodological overview. Understanding the intricate network of connections between neurons is fundamental to comprehending brain functions. A variety of techniques have been developed to trace neural connections, providing insights into structural organization and functional connectivity of the brain. Anatomical tracing methods. These methods involve labeling neurons or their processes to visualize their connections. Anterograde tracing. This technique labels the axons of neurons, allowing researchers to trace their projections to target areas. Commonly used to traces include anterograde transport proteins and fluorescence dyes. Retrograde tracing. This method labels the dendrites and cell bodies of neurons, enabling researchers to trace their afferent connection from source areas. Retrograde traces include fluorogold and rabies virus. Transneuronal tracing. This technique involves labeling neurons in chain, allowing researchers to trace connections across multiple synapses. Rabies virus is commonly used for transneuronal tracing. The functional connectivity methods. These methods measure the temporal connections between the activities of different brain regions to infer functional connections. The functional magnetic resonance imaging fMRI. This technique measures blood flow changes in the brain which are correlated with neural activity. By analyzing the temporal correlations between the activities of brain regions, researchers can infer functional connections. Electroencephalography EEG and magnetoencephalography MEG. These techniques measures the electrical and magnetic fields produced by the brain respectively. By analyzing the temporal correlations between the activities of different brain regions, researchers can infer functional connections. Ganger causality analysis. This statistical method analyzes the temporal relationship between time series data to determine causal influences. It can be applied to EEG, MEG, and fMRI data to infer functional connections. Diffusion tensor imaging DTI. DTI is a type of MRI that measures the diffusion of water molecules in the brain. By analyzing the diffusion patterns, researchers can infer the orientation of white matter tracts, which are bundles of neurons that connect different brain regions. DTI provides valuable information about the structural connectivity of the brain. Challenges and limitations. Tracing neural connection is a complex task with several challenges. The complexity. The brain is highly interconnected network with billions of neurons and trillions of synapses. Tracing all these connections is daunting task. Resolution. Some techniques such as anatomical tracing may have limitations in terms of spatial resolution, making it difficult to identify fine-grained connections. Dynamics. Neural connections are not static but can change over the time in response to experience and learning. Capturing the dynamics of neural connection is challenging. Despite these challenges, the development of new techniques and advancements in computational methods are enabling researchers to make significant progresses in tracing neural connections. These methods are essential for understanding the underlying mechanism of brain function and for developing treatments for neurological and psychiatric disorders. So, in order to study of the living human brain, computerized tomography (CT) scan use of a device that employs computer to analyze the data obtained by a scanning beam of X-rays to produce 2D picture of a slice through the body. Magnetic resonance imaging, the inferior of the body can be accurately imaged, involves the interaction between the radio waves and a strong magnetic field. And next is the fMRI, the functional magnetic resonance imaging. This technique measures blood flow changes in the brain which are correlated with neural activity. By analyzing the temporal correlation between the activities of different brain regions, researchers can infer functional connections. EEG and MEG, electroencephalography and magnetoencephalography, are the techniques measures the electrical and magnetic fields produced by the brain, respectively.